All right, today we're going to take a journey deep, deep inside ourselves. We're diving into this hidden world of microscopic engineering that, believe it or not, holds our entire bodies together. It's this invisible architecture that dictates everything from our shape to how we move. So let's just start with a really basic question.
I mean, seriously think about it. We are made of over 30 trillion tiny individual cells. Each one is its own little world. So what's stopping us from just dissolving into a giant puddle of goo? Well, the answer is this incredible, fascinating combination of a sticky biological glue, a whole series of molecular handshakes, and a dynamic skeleton inside every single one of those cells.
So, to start our journey, we're actually not going to look at a cell. Kind of weird, right? We're going to look at the space between them, because it turns out that space isn't empty at all. Nope, it's packed with this crucial substance that forms the very foundation of all our tissues.
And that substance, it's called the extracellular matrix, or just ECM for short. You can think of it as the biological mortar to our cellular bricks. It's the scaffolding that organizes all those cells into tissues like skin and muscle and bone. And it provides the structural integrity that stops our organs from just collapsing into a disorganized heap. It's absolutely essential.
You can basically think of the ECM as this high-tech, self-assembling construction material, and it's got three key ingredients. First up, you've got fibrous proteins, and the absolute superstar here is collagen. And get this, gram for gram, some types of collagen are literally stronger than steel. It's like the rebar in our bodies, providing incredible strength. Next, you have proteoglycans and GAGs. You've probably heard of hyaluronic acid. Picture these as these giant, fluffy molecules that are masters at trapping water. They create this hydrated, squishy gel that resists compression. And finally, you have adhesion proteins, like laminin and fibronectin, that act like molecular glue, linking the cells themselves to this whole intricate matrix. So honestly, the best way to visualize the ECM is to think of a Jell-O salad. Seriously, you've got the gelatin itself, that's the squishy, hydrated base made of those water-loving gags, and then suspended inside it are those super strong protein fibers like collagen, kind of like the bits of fruit in the salad. This combination creates a material that is both incredibly strong and flexible. It's perfect for building a body.
Okay, so we've got the matrix holding our tissues together. But what about the cells themselves? How do they connect directly to their neighbors? Because, it's not enough to just sit next to each other. They need to form these physical, functional links. And this is where a bunch of specialized connections, or let's call them cellular handshakes, come into play.
So it turns out cells have three main ways of forming these connections. We've got tight junctions, which act like the zipper on a jacket, sealing the space between cells to make a waterproof barrier. Then you've got adhesion junctions, which are more like industrial-strength rivets, powerfully anchoring cells to one another to resist getting pulled apart. And finally, gap junctions, which are like these secret tunnels that allow for direct communication between neighboring cells.
Let's look a little closer at those zippers. Tight junctions are made of these rows of proteins, occludin and claudin, that literally stitch the membranes of two cells together. This creates a seal that nothing can get through. And it's absolutely vital in places like our skin to keep water in and germs out, or in our intestines to make sure nutrients get absorbed through our cells, not just leak between them.
And this is where it gets really real. In a condition like celiac disease, that immune reaction to gluten can actually damage these tight junctions. The zippers in the intestine basically break, making the gut lining porous or leaky. This lets stuff that should stay in the gut leak into the body, which triggers inflammation and all the other awful symptoms. It's a really powerful reminder of just how critical this tiny molecular seal actually is for our health.
Next up, the rivets, adhesion junctions. These guys are all about brute strength. They use these proteins called cadherins to connect to the internal skeletons of the cells next to them. What this does is create this continuous, reinforced network that weaves through an entire tissue, letting it withstand all the stretching and pulling of daily life without tearing apart. This is what gives our skin and our heart muscle their incredible resilience.
And finally, we have the communication tunnels, the gap junctions. These are formed by channel proteins called connexons. You get one from each cell, they line up, and bam, you have a direct portal from one cell to the next. This is absolutely critical in tissues like the heart, where the almost instantaneous flow of electrical signals through these gap junctions is what allows millions of cells to contract in perfect life-sustaining unison.
Okay, so we've explored the world outside the cell and the connections between them. So where do we go next? You guessed it, we're zooming in even further and going inside a single cell. Because every cell has its own complex internal architecture, a dynamic framework that gives it shape and lets it do its job. Welcome to the cytoskeleton. Now, this is not a static, bony skeleton like ours. No way. This is a constantly changing dynamic network of protein fibers. It's the cell's internal scaffolding, yeah, but it's also a highway system for transporting cargo around, and it's the machinery that allows the cell to move and change its shape. It does everything.
So this whole cytoskeleton is built on three main types of filaments. First, you've got intermediate filaments. Think of these as the steel frame of the cell. Their job is pure mechanical strength, resisting stress and anchoring things in place. Second, microtubules. These are the cell's highways. They're rigid tubes that form tracks for motor proteins to haul cargo around. They're also super important for separating chromosomes when a cell divides. And third, actin filaments. These are the cell's muscles, thin, flexible fibers that are essential for any kind of movement or contraction.
Now here is something that is just super clever about how the cell manages all of this. Building these structures costs energy, right? But the cell doesn't just use one type of cache. To control whether it's building highways or building muscles, it uses two different energy currencies. Building the actin filaments, the muscles, is powered by ATP, which is the main energy molecule of the cell. But building the microtubules, the highways, uses a totally different molecule, GTP. This lets the cell independently control these two systems, pushing more resources to building highways when it needs to, and to building muscles when it's time to move. It's brilliant.
All right, time for the grand finale. We've seen the external glue, the cellular handshakes, and the internal skeleton. Let's put all of these pieces together to understand one of the most incredible architectural feats in all of biology, how our muscles actually contract.
Deep inside every single one of your muscle fibers, we find millions upon millions of these tiny repeating engines called sarcomers. It is this beautifully organized arrangement of the actin filaments we just met and a motor protein called myosin. It's the coordinated action of billions of these microscopic engines that allows you to do everything from lifting a weight to running a marathon.
Now, a really key thing to understand about how muscles work is this. Contraction is the active part. It takes a ton of energy. Relaxing, on the other hand, is passive. It doesn't cost any energy. And that's largely thanks to this giant spring-like protein called titin, which gets all compressed during contraction and then just recoils, passively pushing the sarcomere back to its resting state. No energy needed.
So how does this tiny engine actually work? It's a four-step cycle, a little dance. First, get ready. An electrical signal from a nerve releases a flood of calcium. That's the on switch. Second, grab on. The calcium allows the myosin motor proteins to grab onto the actin filaments. Third, pull. This is the power stroke. The myosin heads pivot, pulling the actin filaments toward the center and shortening the whole unit. And fourth, let go and repeat. A molecule of ATP comes in, makes the myosin let go, reset, and get ready to grab and pull all over again. This cycle happens thousands of times a second.
And here's the part that just absolutely blows my mind every time. A single muscle cell, which is thinner than a human hair, can contain over 1 billion of these little sarcomere engines. 1 billion! All lined up, all firing in perfect, beautiful unison to generate the force of one single muscle twitch. The level of organization is just staggering. So, we've gone on this huge journey, right? From the empty space between ourselves all the way down to the molecular motors within them. But let's zoom back out. Why does all of this microscopic architecture matter so much in the grand scheme of things? Well, here's the big picture. Our body is a masterpiece of molecular engineering. This invisible architecture, the matrix, the junctions, the cytoskeleton, this is the fundamental blueprint that governs how we're built and how we function. It's at play in the very first moments of life, shaping a ball of cells into a person, and it's at work every single second of our lives, allowing us to move and think and, well, live. And that leads to one final, really exciting thought. By understanding this blueprint, we're actually learning to copy it. From creating artificial tissues for regenerative medicine to designing new technologies inspired by how our bodies work, we are right on the cusp of using nature's own architectural principles to build the future. The question is no longer just how we are built, but what can we build next? It's a question that started with a single cell, and its answer could truly shape our future. Thanks for exploring this amazing architecture with me.